You're going to see in this video different stages, movement, feeding, and development, in the life cycle of the Myxobacterium Stigmatella aurantiaca. The myxobacteria are gram-negative gliding bacteria. Here you see Stigmatella on the agar. It's about 5 microns long. In their natural habitat, these bacteria use as their source of nutrient other bacteria that they digest by secreting lytic enzymes. And when the nutrient is depleted, the myxobacteria aggregate into numerous aggregation centers consisting of hundreds of thousands of cells. And each aggregation center then develops into a fruiting body, a multicellular fruiting body, which can be from 1/10 or 2/10 of a millimeter tall. As you will see, the Stigmatella fruiting body consists of a stalk supporting several sporangioles. Inside of each sporangiole, the cells convert to resting cells called myxospores. And when the nutrient is present again, the myxospores inside of the sporangioles germinate to give rise to a small population of cells called a swarm. Now you can see the cells gliding on agar. They glide very slowly, and this has been speeded up 5 times in order for you to see them gliding. And here is another view. Not all the cells are gliding, but they will glide later. They periodically stop and start gliding. Notice they seem to glide in groups, which is common for myxobacteria, and it looks actually like a stream of cells. These films were taken in slide culture. Now you can see there are some cells that weren't moving before at the bottom that are moving now. They tend to move in groups, as I say, and here they're moving in circles, which is also common for myxobacteria. They tend to move in groups in circles, in circular motility. These bacteria are on non-nutrient agar right now, but close by there's a source of nutrient, the bacteria called Micrococcus luteus, and later you will see them eating Micrococcus luteus. More gliding motility. The mechanism of gliding motility is unknown. It requires a solid surface, and the cells can move either forwards or backwards. And here you see pictures of a scanning electron micrographs of the various stages in fruiting body formation. In A, what you see are aggregates consisting of hundreds of thousands of cells. And in B, the cells within the aggregates have started to form a stalk. C is a later stage, looks like a mushroom. And in D, the top of the mushroom-like structure has divided into several sporangioles, which you can see on top of the stalk. And what you cannot see is that in each of these sporangioles there are thousands of myxospores. This is a resting stage for these organisms. And if the fruiting body is dispersed to a new location where nutrient is present, then the cells within the sporangioles will germinate, and a small population of cells will emerge. And here you see a fruiting body of Stigmatella aurantiaca. You can see the stalk supporting several sporangioles on top. And what I have done is to remove one of those sporangioles and placed it on agar, and you will see it germinating, and when the cells come out, you'll see them gliding, and eventually you will see them approaching Micrococcus luteus and lysing them and eating them. As I say, this is about 1/10 of a millimeter tall, and they are orange in color. This particular strain was isolated from decaying wood on the campus of Indiana University. This is a top view of the fruiting body, and you can see multiple sporangia, but you cannot see the stalk. They are supported by a stalk. Here is another view from the side, and you can see that the stalk is bifurcated here, so that there are actually two stalks, which is unusual. And here are some sporangioles which were isolated from the fruiting bodies and placed with a fine-tipped glass rod on the aga. Here is a sporangiole, and it is about to germinate. We will be speeding this up 50-fold now so that you can see the cells coming out. Each of these sporangioles is covered by a hard coat which must be dissolved by enzymes secreted by the cells. And now you can see the bacteria exiting the sporangiole. They've already germinated, obviously, and they're coming out as a small population. We call populations of myxobacteria swarms. So a small swarm is coming out, in fact, in more than one place from this sporangiole. See in the top and in the bottom. And this is the latest stage. You can see there are actually two sporangioles that were germinating. And from each one, a swarm comes out. In this particular scene, you can see these light lines in between the cells. It's a better view. These are called slime trails. The myxobacteria make these slime trails while they're gliding, and they glide in the trails. Here is another view of a beginning germination sporangiole. It's only 5 times speeded up in this particular one. That's why the cells do not seem to be moving so fast. Here you can see them moving faster now. In their slime trails. And here is some circular motility. And again. Now we see the myxobacteria approaching the Micrococcus luteus. Micrococcus luteus is on the left. These are spherical bacteria. And the swarm of myxobacteria is approaching. And you will see them approach the Micrococcus and lysine the cells. See the tip of the swarm at the right is moving towards it. Notice how the myxobacteria tend to stay in groups. Now we see at a later time where they appear now to be aware of the presence of the Micrococcus. And pretty soon the Micrococcus will be dead meat. They always seem to move faster as they get closer to the Micrococcus. And Now, in this and in several other scenes, you will see that Stigmatella seems to make a direct approach to the micrococcus. You might assume that they're detecting some chemical or something secreted by the micrococcus, but that is not necessarily the case, because in separate experiments it was seen that they also approached polystyrene beads, indicating that it's possible that the myxobacteria are detecting some stress factors in the agar that were introduced by the micrococcus. Now you can see large groups of cells moving in. Whether they also detect some chemicals released by the lysing Micrococcus luteus is not known. Again, this is speeded up 5 times. Now they're into the Micrococcus colonies now. Notice that the rate of movement is variable. Some cells move fairly rapidly and other cells move hardly at all. Single cells tend not to move. Groups of cells move better. Here we will see again the Stigmatella aurantiaca moving towards the microcolonies of Micrococcus, see, where they will lyse the cells. And at some point, perhaps even before lysis is complete, they will then go to another microcolony of Micrococcus. It's been asked whether or not the cells are following some leader cell at the tip of the swarm, and this is a possibility, but there is no strong evidence for it as yet. When the Micrococcus lyses, then they become phase light. They're not dark or refractile anymore. Here we see some tips of swarms leaving the Micrococcus and going towards other colonies. And it appears when they do this that they do not go directly towards the Micrococcus until they're sufficiently close to it. Then they zip right in. Sometimes it appears that there's a physical attraction, because if you look carefully, it appears as if some of the cells are being pulled in sideways and not actually gliding in. Like right now. It seemed to have been just pulled in. The myxobacteria are enjoying themselves, eating the Micrococcus, going towards the next microcolony. See, that cell just zipped right in. Again, almost sideways. Some seem to be gliding in very rapidly, and some seem to be pulled in. When all of the micrococcus is lysed, if the cell density of Stigmatella is sufficiently high, then the cells will aggregate, form many multicellular aggregates, and develop into fruiting bodies. And within the fruiting bodies, and in fact within the sporangioles, each cell will convert to a resting cell called the myxospore.